Bismillahirrahmanirrahim. Quote of the day. Today is your opportunity to build the tomorrow you want. Today, the topic of my presentation is type of vaccines, and I am presenting it under supervision of Professor Dr. Sarah Afzal. Learning outlines of my presentation are introduction to vaccines, different types of vaccines, and examples of each type of vaccines, recent updates, and the last MCQs. Learning objectives. By the end of this presentation, participants would be able to describe different types of vaccines along with examples. Now, what is vaccine? A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbe, its toxins, or one of its surface proteins. The agent stimulates the body's immune system to recognize the agent as a threat, destroy it, and to further recognize and destroy any of the microorganisms associated with that agent that it may encounter in future. Vaccines can be prophylactic, that is to prevent the effects of a future infection by natural wild pathogen, or therapeutic, that is to fight a disease that has already occurred, such as cancer. Now, types of vaccines. There are several types of vaccines, including inactivated vaccines, toxoid vaccines, subunit, recombinant, polysaccharide and conjugate vaccines, live attenuated vaccines, messenger RNA vaccines, and viral vector vaccines. Now we are going to discuss each one by one. First is live attenuated vaccines. These are the vaccines that contain whole bacteria or viruses which have been weakened or attenuated so that they create a protective immune response but do not cause disease in healthy people. For most modern vaccines, this weakening is achieved through genetic modification of the pathogen, either as a naturally occurring phenomenon or as a modification specially introduced by scientists. These tend to create a strong and lasting immune response and include some of our best vaccines. And live vaccines may not suitable for people whose immune system does not work, that is, who are immunosuppressed either due to drug treatment or any underlying illness. This is because weakened viruses or bacteria could in some cases multiply too much and might cause disease in these people. Now, the most common types of live attenuated vaccines are examples for each are measles, mumps, rubella, varicella for chickenpox, and influenza and rotavirus vaccine. Now, advantages and disadvantages of live attenuated vaccines. Advantages. Infectious microbes can stimulate generation of mammary, cellular, as well as humoral immune responses. Since these can multiply in the host, fewer quantities must be injected to induce protection. A single administration of vaccine often has a high efficacy in produce long-lived immunity. Multiple booster doses may not be required in this vaccine. Whole microbes stimulate response to antigens in their natural conformation. They raise immune response to all protective antigens. Some live vaccines can be given orally. Such vaccines induce mucosal immunity and IgA synthesis, which gives more protection at the normal site of entry. Oral preparations are less expensive than giving injections. They can lead to elimination of wild type virus from the community. Disadvantages. They may very rarely revert to its virulent form and cause disease. Live vaccines cannot be given safely to immunosuppressed individuals. Administration of live vaccines to people with impaired immune function can cause serious illness or death in the vaccine recipient. Since they are live and because their activity depends on their viability, proper storage is critical in this vaccine. Now moving on to second tab, that is inactivated vaccines. These are the vaccines which are created by inactivating a pathogen, typically using heat or chemicals such as formaldehyde or formalin. This destroys the pathogen's ability to replicate but keeps it intact so that the immune system can still recognize it. Inactivated is generally used rather than killed to refer to viral vaccines of this type, as viruses are generally not considered to be alive. Because killed or inactivated pathogens can't replicate at all, so they can't revert to a more virulent form capable of causing. Vice versa of live vaccines. However, they tend to provide a short length of protection than live vaccines and are more likely to require boosters to create long-term immunity. The example for inactivated or killed vaccines are polio, IPV, and hepatitis A. Advantages of killed vaccine that it is safe to use and can be given to immunodeficient and pregnant individuals. Cheaper than live attenuated vaccine and storage is not as critical as live vaccine. Disadvantages. Since the microorganism cannot multiply, large numbers are required to stimulate immunity, so multiple periodic boosters are given to maintain immunity. Only humoral immunity can be induced. Most killed vaccines have to be injected. Some vaccines, such as Bordetella pertussis, induce ill effects like post-vaccinal encephalomyelitis. Anaphylactic reaction to neomycin streptomycin may occur in these recipients. Anaphylactic hypersensitivity to egg may occur in recipients of influenza vaccine. Inactivation, such as by formaldehyde in the case of SARS vaccine, may alter antigenicity, and presence of some inactivated microbes can lead to vaccine-associated disease. Toxoids. Some bacterial diseases, as we all know, that they are not directly caused by bacterium itself, but they are caused by the toxin produced by the bacterium. Like in tetanus, its symptoms are not caused by the Clostridium tetani bacterium itself, but by a neurotoxin it produces, that is tetanospasmin. So immunizations for this type of pathogen can be made by inactivating the toxin that causes disease symptoms. And this can be done by a treatment with the chemicals such as formalin or by using heat or other methods. Immunizations created using inactivated toxins are called toxoids. They are actually considered killed or inactivated vaccines, but are sorry, here comes viruses, but are sometimes gives their own category to highlight the fact that they contain an inactivated toxin and not an inactivated form of bacteria or virus. Example for it is the diphtheria, tetanus, which is a part of DTaP combined immunization. Advantages of toxoid vaccines is that product is devoid of live organism and it implies greater safety. Disadvantages are that multiple doses typically required, relatively expensive to manufacture, cultivation of a pathogen for toxin production is required. Now, other type, recombinant protein vaccine. Recombinant vaccines are made using bacterial or yeast cells to manufacture the vaccine. A small piece of DNA is taken from the virus or bacterium against which it is required to protect and inserted into the manufacturing cells. For example, to make the hepatitis B vaccine, part of DNA from the hepatitis B is inserted into the DNA of yeast cells. These yeast cells are then able to produce one of the surface proteins from the hepatitis B virus, and this is purified and used as the active ingredient in the vaccine. Now, advantages of these vaccines are that those vectors that are not only safe but also easy to grow and store can be chosen. Antigens which do not elicit protective immunity or which elicit damaging responses can be eliminated from the vaccine. Examples are like cholera toxin A can be safely removed from cholera toxin. Disadvantages are that since the genes for the desired antigens must be located, cloned, and expressed efficiently in the new vector, so the cost of this production is very high. When engineered vaccinia virus is used to vaccinate, care must be taken to spare immunodeficient. Subunit vaccine. Most of the recombinant vaccines are subunit vaccines which do not contain any whole bacteria or viruses at all. Instead, these kind of vaccine contain polysaccharides or proteins from the surface of bacteria or virus. These polysaccharides or proteins are the parts that our immune system recognizes as foreign, and they are referred to as antigens. Even though the vaccine might only contain a few out of thousands of proteins in the bacterium, they are enough in themselves to trigger an immune response which can protect against the disease. Conjugate vaccine. Conjugate means connected or joined with some bacteria. To get protection from a vaccine, we need to train the immune system to respond to polysaccharides rather than proteins. But in the early days of polysaccharide vaccine, it was found that they did not work well in babies and young children. So researchers discovered that they work much better if the polysaccharide was attached or conjugated to something else that creates a strong immune response. In most conjugate vaccine, the polysaccharide is attached to diphtheria or tetanus toxoid protein. The immune system recognizes these proteins very easily, and this helps to generate a stronger immune response to polysaccharides. Non-product information sheets, the diphtheria toxoid is often called CRM197 carrier protein because it is almost the same as diphtheria toxoid but not quite. Example for subunit or conjugate vaccine is hepatitis B, influenza, H influenza type B, pertussis, pneumococcal vaccine. Now, subunit vaccines advantages, they can safely be given to immunosuppressed people. They are less likely to induce side effects. Disadvantages are antigens may not retain their native conformation, so that antibodies produced against the subunit may not recognize the same protein on the pathogen surface. Isolated protein does not stimulate the immune system as well as whole organism vaccine. Virus-like particles. This is another type of vaccine which are the molecules that closely resemble viruses but are non-infectious because they contain no viral genetic material. They can be naturally occurring or synthesized through the individual expression of viral structure proteins which can then self-assemble into the virus-like structure. In some cases, the antigens in virus-like particle vaccine are the viral structure protein themselves, and alternatively, they can be manufactured to present antigens from another pathogen on the surface or even multiple pathogens at once. As each virus-like particles have multiple copies of an antigens on its surface, it is more effective at stimulating an immune response than a single copy. However, in some cases, the structural proteins of the virus-like particles can act as adjuvants, helping to strengthen the immune response to the primary target antigen. Example, a handful of virus-like particle-based vaccines are currently used worldwide, which are hepatitis B vaccine and HPV, human papilloma virus vaccine. OMV vaccines. OMV stands for outer membrane vesicles, which are naturally produced by bacteria and are essentially a bleb of the bacterial outer cell membrane. This contains many of the antigens found on the cell membrane, but it is a non-infectious particle. In the lab, these OMVs can be harvested from bacteria to use as vaccines. The OMVs can also be modified so that toxic antigens are removed and antigens suitable for stimulating an immune response can be kept. They also naturally act as adjuvants. Example, this is a new vaccine technology, so there are few licensing examples, one of which is meningococcal B vaccine. Then comes nucleic acid vaccines. These work in a different way to other vaccines in that they do not supply the protein antigen to the body. However, they provide the genetic instructions of the antigens to cells in the body, and in turn, the cells produce the antigens, which stimulates an immune response. These vaccines are quick and easy to develop and provide significant promise for the development of vaccines in the future. Now, one of it is RNA vaccine. RNA vaccines use messenger RNA inside a lipid membrane. This fatty cover both protects the messenger RNA when it enters the body and also helps it to get inside the cells by fusing with the cell membrane. Once the messenger RNA is inside the cell, machinery inside the cell translates it into the antigen protein. And this messenger RNA typically lasts a few days, but in that time, sufficient antigen has been made to stimulate an immune response. Then it is naturally broken down and removed by the body. The RNA vaccines are not capable of combining with the human genetic code, that is DNA. Examples, there are two RNA vaccines authorized for emergency use in the UK at present, the Pfizer BioNTech and Moderna, which are globally widely used, that is for COVID-19 vaccines. They both use RNA vaccines. DNA vaccines. DNA is more stable than messenger RNA, so does not require the same initial protection. They are typically administered along with a technique called electroporation. This uses low-level electronic waves to allow the body cells to take up the DNA vaccine. DNA must be translated to messenger RNA within the cell nucleus before it subsequently be translated to protein antigens which stimulate an immune response. However, there is no currently licensed DNA vaccines, but there are many in development. Now, viral vectored vaccines. As with nucleic acid vaccine, this is a new technology using harmless viruses to deliver the genetic code of target vaccine antigens to cells of the body so they can produce protein antigens to stimulate an immune response. They are grown in cell lines and can be delivered quickly and easily on a large scale. Viral vector vaccines are significantly cheaper to produce in most cases compared to nucleic acid vaccines and many subunit vaccines. There are two types, replicating and non-replicating. In replicating, viral vectors retain the ability to make new viral particles alongside delivering the vaccine antigen when used as a vaccine delivery platform. As with live whole pathogen vaccines, this has the inherent advantage as a replicating virus that it can provide a continuous source of vaccine antigen over an extended period of time compared to non-replicating vaccines and likely to produce a stronger immune response. A single vaccine may be enough to give protection. The replicating viral vectors are typically selected so that viruses themselves are harmless or attenuated whilst they are infecting the host. They cannot cause disease. Despite this, there is still viral replication going on. There is an increased chance of mild adverse reactions with these vaccines. A vaccine to prevent Ebola called Ervebo. It uses a recombinant vesicular stomatitis virus. This vaccine was approved across Europe for use in 2019 and has been used in multiple Ebola outbreaks to protect over 90,000 people. And it has primarily been using ring vaccination where the close contacts of an infected person are vaccinated to prevent the virus from spreading. Then comes the another tab that is non-replicating. Non-replicating viral vectors do not retain the ability to make new viral particles during the process of delivering the vaccine antigen to the cell because key viral genes that enable the virus to replicate have been removed in the lab, and it has an advantage that the vaccine cannot cause disease and adverse events associated with viral vector applications are reduced. However, vaccine antigen can only be produced as long as the initial vaccine remains in the infected cells. This means that the immune response is generally weaker than the with replicating viral vectors, and booster doses are likely to be required in this type of vaccine. A viral vector vaccine developed to prevent Ebola was licensed for use by the European Medicine Agency in July 2020. Oxford AstraZeneca COVID-19 vaccine, which was approved for emergency use by MHRA in December 2020, also uses a non-replicating viral vector. Now, types of the COVID-19 vaccines. Currently, there are three main types of COVID-19 vaccines that are approved or authorized for use and that are undergoing large-scale phase 3 clinical trials. CDC recommend that people who are starting their vaccines or getting a booster dose get either Pfizer or Moderna. However, which vaccine we can get depends on our age. The messenger RNA vaccines are preferred over Johnson & Johnson COVID-19 vaccine in most circumstances. And these are the three main commonly used types of the COVID-19 vaccine. First is messenger RNA, which is very commonly used globally, under which comes the Pfizer and Moderna. It contains material from the virus that causes COVID-19 that gives our cell instruction from how to make a harmless protein that is unique to the virus, as we have already discussed. Then comes the protein subunit vaccine, and the vaccine under this is under development, that is Novavax. And in this, once vaccinated, our bodies recognize that the protein should not be there and build T lymphocytes antibodies that will remember how to fight the virus that causes COVID-19 if we are infected in future. Then comes the vector vaccine, in which comes Johnson, Johnson and Janssen. And AstraZeneca is also vector vaccine. Now, recent updates. New vaccines in development. According to Vaccines Europe, vaccination was first pioneered in Europe, and ever since Europe has been at the heart of the global vaccine industry, playing a key role in research and development of vaccines. Emerging new technologies and research, such as the use of recombinant DNA, have resulted in exciting new breakthroughs and possibilities for the use of vaccines. There are many new vaccines currently being researched and developed within the industry while using of viral vector vaccines, non-replicating vector vaccines, RNA vaccines, DNA plasmid technology, and immunotherapy. And a number of vaccines are in development target diseases that are difficult to treat, such as HIV/AIDS and other viral diseases, cytomegalovirus, dengue, Ebola, cancer, Alzheimer's disease, rheumatic disorders, bacterial disease, and cancers and others. Uh, and several combination vaccines offer practical advantage that can boost vaccine uptake, thereby further improving public health. This was an article titled "Vaccination Against Ebola, one of the most significant leaps in vaccine development and collaboration in the last 30 years." According to it, since Ebola was discovered in 1976, and there have been around 30 significant outbreaks in Africa, the 2014 to 16 epidemic alone claimed more than 11,000 lives across West Africa and had a devastating impact on families, economies. At the time, there was little to stop it. There were no effective treatments, approved vaccines, or even widely available point of care tests. In 2018, Ebola reared its head again in the Democratic Republic of Congo, in the world's second largest outbreak, causing more than 3,000 cases and 2,000 deaths. So today, two different vaccines have been deployed. In November 2019, the global biopharmaceutical company's single dose, that MSD single dose vaccine offering was approved in the European Union. And a few months later, in July 20, Janssen's two-dose regimen was approved in Europe. Novavax, this is a company which is working to produce the subunit vaccine for COVID-19, and it says that it could start making Omicron specific vaccine in January. Now moving on to multiple choice questions. MCQ number 1. Live attenuated vaccines contain whole bacteria or viruses which have been weakened so that they create a protective immune response, not causing a disease in healthy people. Which of the following is not the example of a live attenuated vaccine? Tetanus vaccine, MMR, varicella, influenza vaccine.
A.
Yes, tetanus vaccine, because it is a toxoid vaccine, not a live attenuated vaccine. MCQ number 2. A subunit vaccine contains a fragment of the pathogen and elicits an appropriate immune response. Subunit vaccine is all except a whole purified virus, a purified part or pieces of the antigen, an expensive type of vaccine, a hepatitis B vaccine.
A.
Yes, A, because subunit vaccine is not a whole purified virus, rather it's a purified part or piece of the antigen. MCQ number 3. Which of the following type of vaccines do Johnson & Johnson and AstraZeneca belong to? Live attenuated messenger RNA, viral vector, toxoid vaccine.
C.
Yes, C. We just recently studied this. Yes, C. Johnson & Johnson and AstraZeneca belong to viral vector vaccines. MCQ number 4. Which of the following statement is incorrect about the live attenuated vaccine? It is prepared using whole weakened living bacteria or virus. It can generate a long-term immune response with the administration of a single dose. Measles, mumps, rubella, and oral polio vaccine are live attenuated vaccines. It is stable at normal room temperature.
D.
Yes, its maintenance of temperature is critical in live vaccines. It is generally not stable at normal room temperature because of presence of virus particles. MCQ number 5. It has been reported that thousands of women worldwide are affected by cervical cancer, and equal number of men contract STDs annually. Human papilloma virus, the common microorganism that is associated with cervical, vaginal, and anal cancers, can be protected by the vaccine, which is live attenuated, killed, toxoid, virus-like particle. Any idea?
D.
Yes, right answer is D, virus-like particle. MCQ number 6. All of the following vaccines are examples of a recombinant type vaccines that are currently used in developed countries except a vaccine for hepatitis B, vaccine for human papilloma virus, vaccine against H influenza type B, and vaccine against hepatitis A. The right answer is D, the vaccine against hepatitis A, because it is a type of killed vaccine. MCQ number 7. Killed vaccines are made by destroying the pathogen's ability to replicate but keeping it intact so that immune system can recognize it. Select the incorrect statement regarding the inactivated type of vaccines. Prepared by using heat or radiation for the inactivation of toxins of the microbe. Most effective bacterial pathogen but less on viral pathogens. Poliovirus and hepatitis A are common inactivated vaccines prepared by using heat or radiation for the inactivation of microbe.
B.
No. The right answer is A. Because toxoids are prepared by heating or radiation or for the inactivation of toxins, not the because for the preparation of inactivated or killed type of vaccine, generally they are prepared by heat or radiation for the inactivation of the microbe, not the toxin. MCQ number 8. Ebola virus disease is a contagious disease and outbreaks of the disease have increased in higher numbers to the African region since it was first diagnosed. When did the FDA authorize and approve the first vaccine against the Ebola virus? 2021, 2018, 2010, or 2019?
D.
Yes, D is the right answer. MCQ number 9. Before the invention of polio vaccine in 1955, poliomyelitis was a serious viral infection. Thousands of children got infected and died around the world. Select the incorrect statement about the polio vaccine. It is only present in the form of killed or inactivated vaccine. Live virus vaccine against polio virus can be given orally. The doses of vaccine must be given before children turn 6 years of age. It is present in two forms, oral polio vaccine and activated polio vaccine.
A.
Yes, right answer is A. MCQ number 10. Outer membrane vesicle OMVs are naturally produced by bacteria that contains antigens, although being non-infectious. These can be harvested from bacteria to use as vaccines. The only vaccine using this newer technology is for hepatitis A, COVID-19, human simplex virus, meningococcal B.
D.
D is the right answer, meningococcal B. This is the key of MCQs. These are the references. Thank you.
Thank you very much, Dr. Meha. Again, a very nice presentation. All the points have been nicely presented in a logical sequence. And as we go forward, we are having much clarity about the mechanisms and how the vaccines they are being prepared. And this is very important, especially while we are dealing with COVID-19, with cervical cancers, and other similar diseases. Even hepatitis B vaccine and other vaccines that are being developed through conjugate process and the influenza vaccine to decrease the twin dynamic using subunits, we should be very clear that what are the different processes that are involved in vaccine development. And as you have presented very well, that the vaccines may contain live viruses and they may be attenuated, like they may be weakened or altered so that they may not cause the disease. And this is the point that has to be highlighted for the public health among the masses. There are many people who still think that the live viruses can mutate and they can cause disease. So there are many myths, disinformation that is associated with the vaccine, because the people they don't have the actual idea that how they are weakened, how they are altered, and how they are inactivated. Similarly, the killed vaccines, we in again in COVID-19, the killed vaccines like Sinopharm and Sinovac and CanSinoBio. All these vaccines have been found very effective. Similarly, where the toxins are involved, as Dr. Meha has correctly presented, there are many bacterial diseases like diphtheria and tetanus, where we have the toxoids, and their inactivated toxins can generate a very strong immune response and decrease the illness and the outbreaks of that illness. Recently, we have seen before COVID that we suffer with the outbreak of diphtheria, especially in Lahore. And similarly, few years ago, we have the illnesses that were very much like the different important vaccine preventable diseases. So all these diseases, they are highly prevented if the whole mechanism of vaccine development, from the live attenuated and how the processes are done to make the virus weak or the organism weak, and to alter the organism to find inactivated vaccines. And there are now the mRNA vaccines along with the other subunits that have been used. There are many conjugate vaccines in use like hepatitis B, influenza, Hib, that is the Haemophilus influenza type B. We also have many other combined vaccines like pneumococcal, meningococcal, etc. There are many live vaccines, as Dr. Meha has presented, like we have MMR, zoster, yellow fever. Similarly, we have killed vaccines like rabies. And one of another important type of the conjugate vaccine, and the virus-like particles and the subunits that have been used. And but we are still looking forward for having the campaign for for vaccines against human papilloma virus. Recently, we are working on a cervical screening for the province of Punjab. Later on, we would extend it to whole of Pakistan, and we have seen that there is paucity of information about the cervical cancer prevention, especially against human papilloma virus, and even the vaccine. We know the Cervarix that was available previously against human papilloma virus, especially 16 and 18. It has not been easily available since many years. And another vaccine that is that is very important for the prevention of 13 important human papilloma viruses. Again, that is not available. So we need advocacy. We want more people to spread the importance of vaccines, and to spread this that these vaccines are absolutely safe. And we have seen in COVID-19 that how this disease has been prevented, and how the severity of the disease, the hospital admissions, the ICU admissions have been prevented by the use of vaccine. And this evidence should be used for advocacy of the other vaccines too. I have shared one of a very important information regarding COVID-19 vaccine in the postgraduate group. I hope in further presentations, you would also present certain flow charts and other things. Overall, a very good work, Dr. Meha. Very well done.
Thank you, ma'am.
As there are no other questions in the chat box, so we can stop here.